All right, this is part 2 of chapter 20. We're talking now about PCR polymerase chain reaction, developed some, I guess, 30 plus years ago now to amplify specific copies or specific segments of DNA. It incorporates primers. The primers provide available 3' OH from which to attach new nucleotides, much like we saw in DNA replication. And it also uses a very special DNA polymerase that was discovered from Thermus aquaticus, which is a archaebacterium that lives in hot springs, and therefore its DNA polymerase can withstand very high temperatures, and it doesn't denature at those high temperatures. It's heat resistant, and thus during the sequence of PCR incorporates cooling and heating cycles over and over and over to get DNA to bind and reattach and amplify and so on. But we need to make sure that the polymerase doesn't denature at high temperatures. So this, what we call Taq polymerase, TAQ, is used in PCR because it's heat resistant. So here's the basic procedure. You have your target sequence, and over a series of cycles, which we won't get into overly specifically, you'll learn that in class separately, you anneal or attach primers to the specific segment of DNA that you want to copy, and then you use the Taq polymerase with free-floating nucleotides that are in your solution to add nucleotides one at a time, boom, 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 starting at the 3' end of the primer. And then you heat it up, and by heating it up you get these double strands to break apart into single strands again, so now you have 4 single strands, and then you do the cycle again where you add primers, add nucleotides, add Taq polymerase, you get amplification, adding this second strand, and now you have 4 strands. And eventually, once you get into the 3rd cycle and you have 8 molecules, just your target sequence without any additional DNA will be found in 2 of those 8. And as this process continues through this amplification, by the end of some 20 or 30 cycles, which you usually run it for, you'll have something like 1 billion more of these sequences that you're looking for compared to all the other ones. So we just kind of ignore the other ones because they're very minor or very small in number. Okay, restriction fragment analysis. So this is basically running gels or gel electrophoresis using restriction enzymes and cutting DNA and so on. So this is basically used for comparative studies, comparing one fragment of DNA or section of DNA to another in individuals, in different cells, or whatever it is. And electrophoresis works by taking your DNA and you cut it up with restriction enzymes. We'll talk about that in just a bit. And then you add your DNA to the cathode, to the negative side. Here there's little wells, and you use a pipette man to add them in. And then you attach here this electricity to a power source, you turn it on. DNA is negatively charged, so DNA will run away from the negative, the cathode, and towards the positive anode. And thus it'll move through this gel, almost like Swiss cheese. And the smaller the DNA fragments, the faster they'll move because there's less restriction on them. The larger the DNA fragments, the slower they'll move, so the larger ones will be found closer to the wells. And eventually, after a certain period of time, you get this banding pattern, which they call a DNA fingerprint. And each band is a different length. And you can analyze them or compare this DNA sample to this DNA sample to this one and so on. What you see initially here is not, when you're first running it, this blue is not actually DNA. This is actually loading dye. You can't see the DNA. Not until the very end here, when we look at it under fluorescent light, you see it's fluorescing here. This is from a chemical called ethidium bromide, which is a radioactive substance that very strongly binds to DNA. So only where it sees DNA, it will bind. And then you look under a UV light, and you can see the bands. And the brighter the bands, the more DNA there is in that region. So here we see, for example, comparing normal beta globin to sickle cell beta globin, these 2 alleles from 2 different organisms. DDEL is the name of a restriction enzyme, and in normal beta globin, it cuts in 2 places, so it creates these multiple fragments, 3, 1, 2, 3. In sickle cell, it just cuts once here, that's the restriction site, so you get 2 fragments. So if you run these 2 on a gel, you can see the difference, the sickled allele versus the normal allele. And you can also identify the differences in base pair sizes. This fragment is 376 base pairs, this one is 201, and so on. We'll talk later how you would figure that out. You could then take your gel and do Southern blotting. Now, Southern blotting is a little bit of an old technique. Most people don't do this, or by the time you guys get into a lab in college or what have you, Southern blotting will kind of be a lost art. No one will do it anymore. Most people will just take out the little fragments. For example, let's say they're interested in this fragment from this segment of DNA. They'll cut this out with a razor blade, and they'll put this into a DNA sequencing machine, and they'll sequence it. It'll take an hour. They'll get the exact sequence. They don't need to worry about blotting it and so on. But in any case, the College Board suggests that you should know how to do a Southern blot to identify DNA fragments, so we'll show you how it works. Much like nucleic acid probe hybridization, we have our DNA that we've run on a gel, and we take nitrocellulose paper and we blot it. We put it on top of the gel and we squish it down so that the DNA gets attached to nitrocellulose paper, which attracts it. And then you take that paper, you put it in a bag, and you pour a radioactive probe over the top. Now, your probe is specific for the sequence that you are looking for, so it will bind to the DNA for the sequence that you want. And then you expose the film, and you can see exactly which fragments are the ones that you want. And then you could select those, for example, and do further studies on that. That's Southern blotting. There, of course, is also Western blotting. There is Northern blotting, but we won't get into all those here. RFLPs are restriction fragment length polymorphisms, or differences in DNA sequences between a homologous pair at the same allele. So, for example, the reason why someone might be big A on one chromosome and little a on the other chromosome, or heterozygous, is because of an RFLP, this slight difference. Sometimes, or often, you might find these differences in non-coding regions as well, not just in the alleles themselves. And these RFLPs will create different restriction sites potentially, and thus cut differently, and thus it can be determined by Southern blotting. There's actually thousands of RFLPs present in eukaryotic DNA, and we can use those as genetic markers or locations that distinguish one individual from another. So when, for example, somebody commits a crime and they take a DNA sample, they'll PCR that DNA sample up, and then they'll cut it with restriction enzyme and run it on a gel. They're not going to run the entire individual's genome. They're going to they're going to run the whole genome, but they're going to look for specific sequences that are RFLPs or will distinguish that individual as different from another. They are the genetic markers, and it's known where a variety of these are to locate or to make sure you have the right culprit, for example. Now, the Human Genome Project enabled us to map or sequence the entire arrangement through various techniques. The way that this used to be done was to construct a linkage map through machines, or originally actually it was done by hand, running gels, which is pretty crazy. But you can do a linkage map of only these genetic markers throughout the chromosomes and identify differences. Now, this physical map, of course, was done by cutting the DNA and then overlapping the DNA in different regions where you saw the same nucleotide sequence over and over. And this basically gives you the actual distance between the base pairs. So here we have Dideoxy chain-termination method, which nobody does hardly anymore, where you have your DNA that you want to sequence, and you make it single-stranded, and then you add a primer with polymerase, and you have some free-floating nucleotides, DDNTPs, and then you add some fluorescently tagged dideoxy ribonucleotides. And the basis for using ribonucleotides is they'll still bind, but deoxyribose and ribose, these ribonucleotides don't have a 3' OH like the DDNTPs, and thus once one of these gets added, the sequence stops. And you can then measure that with a laser. You shine a laser through the sample, and it will spit out into this printer the order from left to right, as you can see here, of these labeled strands. So this pink one will show up as pink, and then you have this green one is, we know all the guanines are labeled green and all the adenines are labeled, I guess that's purple, and so on. So based on the color that it spits out, you know the letter, and then you just fill them in, and you're sequencing this strand of DNA. That's dideoxy. There's also Sanger sequencing, which is the similar idea, which he kind of led to this dideoxy, which we won't really talk about. Sanger won the Nobel Prize for that. What was then used is this shotgun method by the Human Genome Project and Celera, a company that was competing with them and then ended up partnering with them in the end, where you essentially blast the DNA into a bunch of little pieces, and then you do this overlap and let the computer do it. So here you have your DNA, blast it into a bunch of different pieces, and then have the computer piece together the overlapping regions into one long strand, and that's your sequencing. And this actually sped up the process significantly, so that we actually were able to finish in 2003. It would have taken much, much longer probably. So now we have all this DNA, we have to know how to use it. We still don't exactly know what all these different sets of genes and so on do or how they work, but we're working on it. So for example, we now know that there's about 25,000 genes. Here is an example of the number of genes in different organisms. So for example, in nematodes, C. elegans, those are little clear worms, they have 19,000 genes. And rice has 60,000 genes. Strange to think that rice has more than double the number of genes that you do, here at 25,000. But in any case, funny to think about that. So we can compare and discover the function of these new genes. We have to determine that. Sometimes we can take an organism and inactivate a gene, so block that gene from being expressed, and then see what happens in the affected cell or the affected organism. These are basically called knockouts. It's pretty typical in mice. You can buy knockouts of every mouse gene that is known from a company. They're not very cheap, and you see mice have all these various different characteristics. This is kind of like the try and see approach. You can also use microarrays to look at gene expression when comparing groups of genes, as we talked about before, whether it be different tissues or different times. I want to look at the gene expression in one of the cells of my eye during the night versus during the day, exposure to light or not, I don't know, making things up here. So here's how microarrays work. You take your tissue samples and you take the mRNA and you create a cDNA library with reverse transcriptase. But this cDNA library you make with fluorescently labeled nucleotides. And then you take the cDNA mixture and you apply it to this slide, this little glass slide with tons of tiny, tiny dimples. Now they're created with millions of little dimples. And you put DNA samples in each one of those dimples, and then you wash the microarray with these probes, this cDNA mixture that is fluorescently labeled. And you rinse off the excess cDNA, the fluorescently labeled cDNA that you used as your probe, and then you scan it for fluorescence. And anywhere where the cDNA library, the cDNA fragments bound to your specific samples of DNA will light up or fluoresce. As you see here, this one and this one. This one didn't have it. It doesn't have our sequence that we're looking at, or a gene that was expressed, for example. And it creates this pattern, this light. This is, for example, a microarray of yeast with 6,400 spots. A computer will then analyze this color pattern to identify and tell you, spit out information of which different well was hybridized to, was basically expressed. And so this is comparative genomics, which is super, super important whether you're comparing species or cell type or what have you. Proteomics is kind of the next thing, where we have the proteome is all the proteins encoded by a genome, and this is an even much bigger project than the Human Genome Project and is currently ongoing. And then just to give you some terminology here, SNPs, or single nucleotide polymorphisms, are differences between organisms where one nucleotide is different within a gene. So even you might consider alleles or RFLPs are like SNPs, differences, and we can identify those differences by running a gel, for example, cutting with a restriction enzyme. It'll cut in a different place, kind of like we mentioned before. So these are SNPs. Lots of benefits to DNA technology, of course, and some fears, of course, but we can identify human genes where there's a mutation that causes a genetic disease. So, for example, cystic fibrosis, we know, is the result of a single nucleotide polymorphism in one gene. And so we use all these various techniques to identify and to learn how to treat genetic disorders of various types, such as the one we saw here with sickle cell anemia, where we have differences in these restriction sites between the disease-causing allele and the normal allele, and thus differences in when you run a gel, and you can identify where the difference occurs, where it resides. How many slides we got left? Let's just push through to the end here. Gene therapy used to be 30 years ago was the next big thing. There were some issues. Someone died during testing, scared away scientists. They left for 10 years, stopped kind of doing gene therapy, and now it's back where you take an afflicted individual's genes and you give them the correct genes, so to speak. So if you're diabetic, you could have gene therapy by having the correct, you kill off all the DNA that is missing or produces non-functional insulin, and you give them the functional insulin gene. Now, of course, this doesn't work exactly like that yet. Still ongoing, but you use vectors to deliver the genes into these cells. Typically, they're retroviral vectors, so you incorporate your clone gene, put it into a virus. That virus will insert it into the host cell for you through this injection, and then the new DNA will become part of the chromosome of the host cell. That's gene therapy. Other things include large-scale production of hormones. We talked about large-scale production of insulin in E. coli through these DNA technological processes. Production of safer vaccines, also used in crime scene cases. We talked about how this, for example, is from the O.J. Simpson case. This is the actual DNA fingerprint of the blood on O.J.'s clothes. This is the victim's blood, and this is the defendant's blood. You'll notice that he did have blood on his shirt. The blood on his shirt did not match O.J.'s own blood. The blood did match the victim's blood. Funny thing is that DNA fingerprinting and PCR was so new that the attorneys were able to convince the jury that there were all kinds of problems and potential downfalls and that you couldn't trust this yet. And the scientists that were trying to explain it were far too smart for any of the jury, and none of the jury really understood what they were hearing. So they told later the media that they actually disregarded all the DNA evidence, where it's quite obviously clear that O.J. had done it. Environmental cleanup, we can modify microorganisms to help clean up potentially toxic waste materials, like in the oil spill in the Gulf just recently from BP. BP, whether they were allowed to or not, is up for debate, but they released genetically modified bacterium that feed off of oil to eat up all the oil that they spilled, or to help they eat and survive off of the oil. They were genetically modified to do that. Or you can use it to improve agricultural productivity, to increase the protein content of rice, for example. They call it yellow rice. Or to make corn not susceptible to certain pests, and thus minimizing the use of pesticides, which would be a good thing. We can create transgenic animals. For example, here we see sheep that have been genetically modified to produce hormones in their milk. For example, growth hormone, which you can then extract from their milk and bottle and package, sold as medicine. They're genetically modified early on, and every time they create milk, it's filled with growth hormone. They're pharmaceutical factories or various other drugs that they can create for you. All kinds of things that you can use through genetic engineering in plants that major companies are doing or working on, like Monsanto and Dow, for better or worse. How would they do this with plants? Well, there's a very special plasmid that they would use. For example, here, this TI plasmid, where they create a recombinant plasmid and then insert it into the plant cells, and now this plant has a new trait. This is actually not all that difficult to do, and many of you could do this for some of your science fair type projects because plants are very easy to use, and you could culture plants in petri dishes, which is also fairly easy to do. Good or bad, it's up for debate, these genetically modified organisms, or the use of genetic engineering on living organisms. That's for you to determine and you to listen and learn as you get older.